Hello everyone, today I will explain the Primer3plus web tool. This explanation is based on the publication in Nucleic Acids Research named Primer3plus an enhanced web interface to Primer3. The purpose of this tool is to design primers, which can then be used for many different purposes. The most important properties in designing primers are first of all the melting temperature, or TM. This is always between 55 and 65 degrees and needs to be approximately the same for both of the primers. This is because when we do a PCR by example, both of the primers need to work at the same annealing temperature. GC content is another property that is important. GC bonds have 3 hydrogen bonds compared to 80 bonds only having 2. This means that a higher temperature is needed to break the GC bonds. Having a GC content that is too high will give a melting temperature that is too high. On the other hand, having a GC content that is too low will give a melting temperature that is too low. Another property we have is self-complementarity, which is when 2 primers bind each other instead of the template DNA. So rather than elongating the DNA, it will just make DNA products of the primers. Lastly, we have the length of the primer. If the primer is too short, it will not be specific enough. This is especially so when we have a very large DNA sample. So the chance is very big that there will be a lot of sequences where the primer can bind. So then we will have a lot of product on our agarose gel that we don't need. The primer can also be too long, and in that case it might not bind because it can't find the sequence. The first and most important task we can do with this tool is detection. So we make primers to detect a specific DNA sequence, and more specifically this is done with PCR, or polymerase chain reaction. Here we have a double-stranded DNA sample, which we first denaturate. Then on these single-stranded DNA, our primers can bind. Then DNA polymerase, an enzyme, will then use these primers as a starting point to build in nucleotides and so copy the DNA. In the second cycle here, the wanted product will be formed, and then in the next PCR cycles, this specific DNA sequence will be copied many times, and then after the PCR we can load it on an agarose gel, and then we should see bands at the expected size. Some other maybe less important things we can do with this tool are first of all cloning. Here we isolate a specific DNA sequence to then ligate in a vector, transform in a bacteria, and so on. So the whole cloning procedure. So in most cases this will be a vector or a plasmid with our wanted DNA sequence inside, and then we can use the primers to isolate it from the plasmid. Another task we can do is sequencing. Here we design primers for sequencing, and sequencing is to determine the exact sequence of a DNA sample. We also have the task primer list. This will give, so when you enter a DNA sequence, this will give every possible primer that can be used for that DNA sequence. And we also have primer check, and in that case it will give information about our primer, like the length, the GC content, and so on. The interface. So the first thing we see here upper left is the task. Here we can select the task I mentioned previously, so detection, sequencing, cloning, primer list, and primer check. Here in this white space we can paste a nucleotide sequence that we get by example from NCBI, or just maybe a sequencing result we got. And in some cases, when you have a FASTA file, you can also upload this. Here we have 3 markers. This is exclude, target, or include. With exclude, you select a region in the nucleotide sequence where the tool cannot look for primers. So it will not look for primers in that region. Then we have target. In that case, it will look for primers that will include the entire region you selected. And then we have include, where it will look for primers in the region you selected. Now target and include may sound similar, but the big difference is that in the case of target, the primers will be outside of the selected region, and in the case of include, they will be inside of the selected region. Here on the bottom, we can also use our own primers. By example, when we did a project and we tested both of our primers and only the left primer worked, we can insert our left primer here, and then it will look for right primers that work with this left primer. Once we have filled in all of these, we can click on Pick Primers. But before we do that, we also have an important tab called General Settings, where we can change some important aspects. The first one is the length of the PCR product. In some cases, this may be important in your project. And so the length of a PCR product is basically the size you will see on your agarose gel. In this case, it's put at random, so it can be any size, but you can specify this. Then the other three things we have here are the things I mentioned earlier that are important in creating primers, and that is first of all the primer size. Here it is set to a minimum of 18, a maximum of 27, and optimally 20. You can also change the melting temperature. Minimum is here 57 degrees and here 63 degrees with optimum of 60 degrees. And then we have GC content, which is between 20 and 80%. We can also change this. Then we will try some examples of this. So first we need a nucleotide sequence, and the best way to get this is to go to NCBI gene, where we can just type in any gene we can use. So by example, insulin from Homo sapiens, so humans. If we go down here, we have insulin Homo sapiens. We click this. Then we go to NCBI reference sequences. And then go down here. We have 4 forms because of alternative splicing, but we will just use the first one. Then we click FASTA file to have the nucleotide sequence, and we can copy this. You can also save this in a text file and then save it as a FASTA file, but for now we'll just copy and paste this. So we have this now here. I will not touch the general settings because in most cases this is good, but if you, by example, have a sequence and it cannot find primers because this is too strict, you can change this if you want to, and then maybe it will find primers. So then we use Pick Primers. And as you can see, it gives us 5 different primer pairs which we can use. So let's just look at the first one. So here, this is the left primer. This is the sequence of the primer. And then we see some information. So here we see that it starts at position 128. It's a length of 20 base pairs, melting temperature of 60 degrees, GC content of 50%. Then we have our right primer, which starts at 372, also a length of 20 base pairs, 60 degrees, and then 60%. So this is good. And then finally we have our product size. So on an agarose gel or DNA sample will be 245 base pairs. So then we go back. So let's test these markers. So let's give by example that we want to isolate our coding sequence. So we go down here and we see that our coding sequence goes from 60 to 392. So how we insert this, we go to target because we want the entire coding sequence. We type first 60 because that's the starting position, and then we type how many nucleotides after it the region goes. So we see here it is 392 minus 60 is 332 plus 1 is 333. And then we pick primers again. So now as you can see, it will actually have a primer here and a primer here. And this green part is the region we selected. So it will make primers outside of that region. So then we go back and by example we put this in included region, then pick primers. You can see that the primers are now inside of the region that we selected. Then we go back to exclude. So let's just say 1 to 60. Then pick primers. As you can see, it will look for primers outside of the region. So this is the region we selected where it cannot look for a primer. As you can see, it's outside of that region. So the other functions in here are cloning, sequencing, primer list, and primer check, but I will not go into detail with these. So finally, I will compare this to another primer creation tool, and that is Primer3plus from NCBI. So let's just copy this sequence and paste it here. So as you can see, we have many similarities between the two tools. You can also type in the range. In this case, you cannot use exclude target or include. It will just use the include one. You can also insert your own primers here, change the product size of your PCR, change the number of results that it will give, change the melting temperature. So what we do have here is that we cannot change the GC content of the primer. So when we then go to get primers, we will also see a very important advantage that this tool has that this one doesn't, and that is that the tool immediately gives the DNA sequences where the primer binds to, because this is connected to a database. So it will show that the primers that we got actually bind to this one, so Homo sapiens insulin, but will also give accidental sequences, so sequences where the primer binds to that may not be wanted. This is important because when you make a primer set that is specific to a species, and then you see that it also binds to the species you don't want it to bind to, you may have to change your primers. So here we now see the results from Primer3plus on the same sequence. I skipped this through the video because it took a long time for it to get this result. So that's another good advantage of Primer3plus, is that it works way faster than this one. Here we see a nice graph of the different primer pairs and where they bind on the nucleotide sequence. Then we see our first primer pair. We see the sequence where the template strand, so plus or minus. We have the length, the start, and where both of them start, where both of them stop, their melting temperatures, GC content. And we see that the product length here is 359. So it's different from the first one we got here, which was 438. So here we could specify it better because we now included the entire CDS, and in this case it might not be like that, or we don't really know actually. We see here, which is an advantage of this one, is that it shows to which DNA sequence it binds. So it only has one intended target, which is good. So it's the insulin that we looked up on the gene database. But then we also have some potentially unintended templates. Here the first three are the transcript variants you saw earlier. But the other ones seem to be predicted sequences from other genes. Then we go down a bit, we can see our second primer pair with again all the information. So how I would use this tool actually is in combination with Primer3plus. So we'll show that as a last thing. So here we look for our sequence, and what we can actually do is, so let me just go back here, copy this, and then we can actually use these primers. And then get the primers. So here we now have the results of using the tools together. We see here again a graph. We only have one primer pair because we inserted our own primers. We see if we compare, we here have indeed 438 base pairs. The start seems to be the same for both. And the TM is slightly different actually. We see that this one is way lower on this program than it is here. So that's also a difference between the two tools. Then here we have the intended targets, which is again the insulin transcript variant 1 we got from NCBI gene. And then here we have potentially unintended templates, and we see that there are significantly less unintended templates than by just using primer blast alone. So that's also better. So then the final conclusion, we see that Primer3plus is a very good tool to design primers. It has many different parameters that we can change to really specify the primers that we obtain. I did not show you all of the parameters, but there are a lot more. So for more experienced lab technicians, it's really useful. We also get our many different primer pairs obtained very quickly compared to primer blast. It was really a lot faster. And I think to use this program and it's best, I think it's best if we use it in combination with primer blast to just see if it does not bind to any unintended targets. And we also see that the melting temperature is slightly different, so maybe that might be a problem later in the PCR also. So thank you. This was my presentation, and thank you all for watching.